Hello everyone, welcome to the next lesson in genetics. Today we're going to be talking about some genetic mutations and sort of the basic idea behind these genetic mutations. I've filled out these notes. They are of course always available on the Google Classroom, and also you can pause and fill them in as you need. So I'm going to start here. I'll sort of highlight as I go to tell you what I'm talking about. So genetic mutations, one of the concepts I want to get across right away is that through the processes of sexual reproduction, we are all born unique relative to any other option that could have possibly occurred in that conception. And this is because of at least two factors in addition to quite a few other factors. But at the very least, we are all unique because of sexual reproduction, and that is because of the first principle, which is crossing over. So through crossing over, we shuffle the homologous chromosomes, and that causes a change in the distribution of alleles in every single gamete. So every single gamete has a unique combination of alleles that is different from all the other gametes, and that's because of this crossing over event. And we can actually talk a little bit more in detail now about this than we could before. We can now think about this as in the parent, the F1 and the F2 generation. So the parent generation, which I drew up here, right? This parent generation gives a gamete. In this case, this individual inherited the two dominant genes A and B from one parent, which have been described in the blue. And at this stage in meiosis, there are two copies of that chromosome through the chromosome duplication and interphase. And then on this side, we have one parent. I just identified red as female and male as blue. Maybe that's too typical of me. And that parent donates a recessive A and a recessive B. And so those two chromosomes come down. They allow that person to live and survive, and they use those chromosomes. They are heterozygotes for both traits because of one parent who donated dominant genes, one parent who donated recessive genes. And now it is time to produce some gametes. So during that first interphase, that first meiosis, prophase 1 stage, there's crossing over, and this shuffles it. So now they have a what do we call it? Sister chromatids, and these sister chromatids are now different. They're no longer identical. Remember, through all of this person's life and all of the other cells in their body, the sister chromatids are always the same because they're always undergoing mitosis. But now that we're undergoing meiosis in these gamete cells, gamete cell formation, we are going to undergo this type of crossover, and now the gametes are starting to get messed up. So now we have an example of one chromatid that is still entirely, this is entirely the male parent, but this gamete here is not identical to the male parent. It's actually identical to the female. It's half female, half male parent. So it is inheriting half the traits of the male parent, half the traits of the female parent. This only applies to homologous chromosomes. So if this person was male and they had an X and a Y chromosome, then there would not be any crossover between those because they're not homologous. But if they were a female, there would still be crossover between the X chromosomes, so that every female child or every female gamete is unique in its X chromosome in addition to being unique in all of its other chromosomes. Males will pass down their mother's X chromosome and their father's Y chromosome, so the inheritance pattern through males is actually going to be kind of boring. There's no crossover happening for those genes on the X and Y chromosomes. There's no change, at least, through crossover. In fact, Y chromosomes have very little difference from one generation to the other because of this case that there are no crossover events. So the Y chromosome can actually be traced all the way back through an entire family line as being the exact same Y chromosome. And there's a lot of interest in what that means and all that sort of things. There was even a study that I read about at one point. I don't remember all the details of it, but there is some section of Europe or something where they can actually look at the Y chromosomes, and they can figure out that the Y chromosomes in that section of Europe have a higher percentage of the profitable and rich elite of the sort of Middle Ages. So in the Middle Ages, men would be, men who are richer would have more sons who could inherit their money. Their sons would not be sent to war and wouldn't die, and those chromosomes would stay. They would be able to pass down their fortune to the next generation. Meanwhile, those who are working in the fields and things, their sons had a higher mortality rate and that sort of thing. So the Y chromosome over time has more of the more rich and profitable family lines in their generation. And additionally, there was another study done on the Y chromosome in, I believe it was in Russia, where they found that the Y chromosome has a huge change in diversity after World War II, where a majority of Russian males were sent to war and did not return. And the Y chromosome and the diversity of the Y chromosome actually changed quite a bit from that event. Anyway, so the Y chromosome is an interesting piece to study, but it is kind of boring in a way, and that boringness of it, the lack of crossover, makes it a useful tool for measuring backwards in time, I guess. Anyways, I'm getting off track. I wanted to make this video short. Okay, so random fertilization is the other aspect. Remember that right here, after the single meiotic event, there are 4 different gametes, and all of them are different. Multiply this out by the billions of different, different lengths of chromosome that you have, and all of the different possible, genes. You have, there are 8 billion sperm produced every 64 days in a male human, and every single one of those sperm are going to be different from all of the others. There's a little bit, the randomization here is a little different in terms of female eggs, because the eggs are not produced in such high quantities. And in fact, this is where one of the places they look for mutations is likely from the male, because the male likely donates the most mutations because their cells are undergoing the most divisions. So if you take this genetic variation together, you get about 8.4 million possible chromosome combinations. So that's and if you multiply that out into a zygote, so combining 2 different gametes together, that gives you 70 trillion different combinations for just 2 people coming together to create a child. So 2 gamete donors coming together to create a child you are one of 70 trillion for that pairing. Now, that makes a massive amount of variation if you include all of the people in the world and all that sort of thing. So there's a lot of options there. Now, I do want to bring up one note up here, and this is something I think I'm going to come back to when we get to the DNA unit, is the concept of linked genes. I'd like to loop the DNA unit into this meiosis conversation later as well. But linked genes are genes that are actually physically close to each other on the same chromosome. So these are genes that are if we have a chromosome, I'm just going to draw one up here. So just even drawing a chromosome like this, if I put genes, and I put, like, say, gene A over here, and it's gene A is like this long, and then we put gene B here, and then we say that gene C is this little tail up the end up over here, and I'm going to be randomly making cuts on this, randomly cutting the chromosome and causing crossover. The chance of me making a cut, somewhere in this region between B and C is pretty high compared to the chance of making a cut somewhere in here. That chance is much lower. So A and B are more likely to be inherited together than C. So if you inherit, so if this is, this is from the male parent or whatever, versus the female parent, then if this section gets put into the final gamete that is used for fertilization, then it's likely they're going to inherit both of those traits from the one parent. And that's one of the things that they will study in really large scale, things like 23andMe. That's one of the things they're looking at. So in a 23andMe test, they're actually looking at your family history. They want to know more about your family history so they can kind of map out which genes are close together on a chromosome based on the inheritance patterns they're seeing and the genetic maps that they're getting from your genes. Okay. So a really common example is red hair gene and freckles. Those genes are typically very close together on a chromosome. And so those people who have red hair, they have a higher chance of having freckles as well. And so, freckles might not develop until later in your life, but if you see someone who is a child, or you see a child with red hair, you might be able to make the prediction that they're going to have freckles sometime in the future because they probably inherited both traits from their parent. Okay, so mutations is the third way that we randomize. And so mutations are inheritable changes in the genetic information, either in the composition and organizations of the chromosome or in changes in the information that they carry. And we'll talk a little bit more about these changes in information. That's going to be more of a topic for the next unit after we've discussed how DNA information is actually organized. But we know about a little bit about what a chromosome looks like, and so we can talk about the larger compositional changes that can happen of a chromosome in sort of the large scale, and then we'll focus in on the small scale changes later on. So, and another concept that we have talked about, which I did not put a little note on about, is that we also know that from our evolution unit, we discussed how all species have a unique mutation rate, which is a driving force for natural selection. And so we did discuss a little bit about how things like bacteria have a slow mutation rate, things that undergo sexual reproduction have a higher mutation rate. And so I think it's in our evolution unit that I mentioned that, humans in general, I believe that we gain about 3 new mutations every generation in our coding regions on average. And so you have 3 unique features that your parents did not have. And where those traits are and what those traits do, they might be something benign, they might be something interesting or new. They might be something that other people have, but yours is a unique mutation. So we're going to talk specifically about the chromosome mutations. These are significant changes in the physical structure of the chromosomes, and these are the 4 ones we'll talk about, and we'll talk about one extra bonus one in a second. So the first one is deletion, and as the name sort of suggests, this is essentially a removal of a gene. It's a removal of a section of the chromosome, and this has a chance of removing one or more genes from the chromosome itself. And this deletion can happen at any point, but when the chromosomes are being duplicated or manipulated, especially when they're chromatin and they're that loose form, that loose form can occasionally loop in on itself, and then an enzyme can come around and sort of lop off this. Let's maybe do this. And they might loop in on itself, and then an enzyme comes around and it goes to repair the gene, and it ends up repairing it in such a way that it ends up lopping off that section, that little loop. And that little loop is lost to time, and it just shortens the chromosome. This can have all sorts of other implications because location is important on a chromosome. So when the chromosome becomes shorter, then location identification can become a problem, and it could cause problems for all of the information that's down the line, because now all of a sudden a piece is missing, and so every piece after that point might be affected as well, depending on how that particular section of the chromosome is read. So this can cause all sorts of interesting things. One is the cri du chat, which is the cat's cry disorder, and this is, I believe, is a syndrome. So that means that it's a collection of various impacts on the individual, of various. They live with a variety of symptoms. Some of them, and in different individuals, the symptoms are more or um, less important, and the the major one that they will first notice is that the infant will often be born with a larynx, an underdeveloped larynx, or a abnormal development of the larynx, which can cause their infant to cries to sound like a cat. That's where the cri du chat stands for cat's cry in French. It also impacts the child in many other ways, including developmentally in some cases, and it can be uh, quite a syndrome to have to exist with, and it's a very simple mutation. I believe it's only the deletion of a few bases of the DNA strand, and then it causes one protein or so to stop functioning, and that causes a whole cascading impacts. And a couple other examples of deletions that are problematic are things like very small deletions like muscular dystrophy is a single base deletion, and I believe it is cystic fibrosis, I believe, is also one. And so these are just very simple, small changes to the chromosome, but it impacts a very particularly important important piece of the chromosome that has important function necessary to development. Translocations are an interesting one. There's a lot of different examples of translocations, but this is essentially a movement of a section of the chromosome from one chromosome to another. And with that movement of a piece of the chromosome, there is a chance that a gene is that there are genes that are moved that contain important information. And as I'd mentioned, the lack of crossover between X and Y, well, there actually is an example called the SYR mutation, which is essentially caused by crossover, an attempt at crossover in the cell between the Y and X chromosome. And because the X doesn't really have information to give away, the Y chromosome can actually donate a piece of itself, or even an entire section, or larger. It's a wide impact, a wide variety of impacts in this type of mutation. And essentially, a piece of the Y chromosome is moved over to the X. The individual now has the XX, might even inherit an XX genotype, but one of those X's essentially has tagged onto it a section of the Y chromosome. And so this can have, in some cases, enough Y chromosome that male traits will develop even though they have the XX genotype. So this is one of the cases where I do want to emphasize that the genetic development of sex of the assigned sex is not necessarily a straight road. It's not as easy as we often attribute it, that if you are XX chromosomes, you must develop female. That's not the case. There has been quite a few examples now where there are individuals who come to realize that they actually carry the XX genotype, but they have one of these types of mutations, and they don't have a Y chromosome. They have some piece of a Y chromosome that has given them male phenotype, but they have the XX genotype. So a very interesting example to explore. In our evolution unit, we also discussed the snake venom case, where snake venom is thought to be or has been studied, and the theory provides that it is a translocation of a stomach enzyme gene. So if you move you move a gene from one chromosome to another, it often will have an impact on what cells are using that piece of information. So a section of gene that was used to make enzymes for breaking down meat in the stomach now are on a chromosome that happens to be more active in salivary cells, so like mouth cells. Those mouth cells now are producing an enzyme that's good at breaking down meat, and over natural selection and time, the snakes find that this is a useful trait for um, catching prey. So an inversion is, as it sounds, it is a chromosome section that is inverted. And so you can see a little diagram of how that would look. If we list the sections A through I, then A through I over here, then an inversion would switch the order of some of that section. And that switch of order and location could have a huge impact. And I don't really have a good example. This is one example that was given to me, saying that inversions have been linked to some forms of autism. Autism, of course, is a spectrum of disorders under which have similar traits, have some similarities in the expressed traits, but the actual cause of those varies from individual to individual. And so one of the causes that has been found apparently is that these inversion mutations can have some impact on the individual that would classify them as living with a form of autism. Duplication and amplification is a pretty common mutation that we see, especially when we compare species through the evolutionary lens. So here we are creating copies of sections of the chromosome on the same chromosome, and this increases the copies of certain genes. Sometimes these copies even get translocated. So for example, those snakes probably still have stomach genes, the stomach enzymes to break down foods inside their stomach, in addition to having the translocated enzyme gene for making venom. This can cause an increase in genetic diversity by multiplying out the amount of, of genes for hair color. For example, you increase the diversity of hair types, of hair color types, and it also might reduce fertility apparently. The Fragile X syndrome is an example that's sort of an extreme example. That's a case where there is actually an extra X chromosome or an extra piece of the X chromosome. This is a little bit on the outside of what I would call a duplication and an amplification. It's, or actually, sorry, the Fragile X is a duplication in the X chromosome. I apologize. And this has a spectrum of impacts on the on intellectual development, I guess. In terms of the evolutionary lens, salivary amylase is a good example of an evolutionary example of duplication and amplification. So there are certain groups of Homo sapiens sapiens who have extra copies of the salivary amylase gene. This is a gene that breaks down starch into sugars. And these are groups that have long cultural traditions of eating high starch diets. So these are rice eating groups of Homo sapiens, bread eating groups, and maize eating groups. So these groups are, the groups that are traditionally sort of groups of humans essentially found that are located in, say, I believe there's people who have genetic ancestries from Eastern Europe area, from certain sections of Asia, and I believe there's a section of Africa as well. It's the western section. There's a map in my head here, and I believe also South America. All of those sections, those groups of individuals with an ancestry there have extra copies of salivary amylase, up to 6 copies, I believe, 6 or more. There might even be 8 copies in some people. And so we might end up doing an activity on this. Yeah, maybe we'll do that activity. I have one or at least I'll have a video that I'll post about this example. It's pretty cool. I think it's very neat. Okay, the last one I want to put in is the chromosome inactivation. So this is a large scale, not really mutation. It's something that happens in all cells, particularly in female cells, is that many of our cells only need one copy of a chromosome to function. They only need, say, one copy of a gene to actually do the function it needs. And so in some cells, you'll have a chromosome inactivation. And one of the chromosomes that this is the case for is the X chromosome. Remember that male cells can develop completely with only one X chromosome. So this tells us that the normal development of human cells actually only requires a single X chromosome. And that, so in females who undergo normal development, only one of the two X chromosomes is active in any cell. And so the other X chromosome ends up inactivating its cell, and they are known as Barr bodies. They were discovered by Dr. Barr, a female scientist who studied cells for many years. A known and celebrated scientist in her area of study. And she identified them in these images here. So you can see that in the first two images, we have Barr bodies here, and I believe here as well. Or that might be just another chromosome, but these they've identified those as the Barr bodies, the dark stained area in the lower proton of the nucleus of A and B. This is the Barr body, so that's one of the X chromosomes. And the other X chromosome is somewhere else in the cell doing its job, unwound, not wound up like these little Barr bodies. They're sort of, the chromosome gets super wound tightly around a bunch of protein and is no longer active until cell division. Now this inactivation is random, so of the two X chromosomes, one of them is randomly active and one of them is randomly inactive. So you can see, for example, in this early embryo, notice that there are 1, 2, 3, 4. We have 1, 2, 3, 4 cells that have the orange and 4 cells that have the dark black color here. And but because it is random, that is only on average that is going to be 4 and 4. There is also the possibility that you could have all of them be orange or all of them be black. They would be less likely, but it is still a possibility. Notice that in the male cell here, that it has no Barr body because the male cell only has one X chromosome. It needs it to stay active, it needs it to stay unwound. One of the things that came out of this is this little box I've put here, which is a newly discovered type of intermediate color blindness that some females have reported. And this is because they might be heterozygous for color blindness. And that means that their retinal cells have a 50-50 chance of being colorblind. Some of the cells that are forming their eyes are going to have the one Barr body, and some of them are going to have the other Barr body. And if it's the normal cells on average that are if it's normal X chromosomes or X chromosomes carrying the normal allele that are more common, then they'll have relatively normal color vision. If it happens to be, because it is a random event, if it happens to skew towards the color blindness side, then they might have some of that color blindness. And there was a study, and I believe the conclusion was that they did find that there was the heterozygous females for color blindness did have a preference for bold color palettes, like wearing lots of reds with greens or yellows with purples, and or painting their houses in certain ways, or preferring certain types of flowers, because, and they theorized, at least in this paper that I read at that time, whether I could find this reference again now, that this variation of color blindness might mean that they prefer those bold colors, because those are typically easier for colorblind people to see, is when the colors are very extremely different. Calico cats is an example also of this sort of made visible in the case of the calico cat, the black and orange colors of the cat are carried on the X chromosome. So males can only be black or orange. So here's my black male and here's my orange male, and that's because they only carry one copy of the X chromosome, so they only carry either an orange gene or a black gene. And meanwhile, females can be, who are homozygous for black, will of course be black, homozygous for orange will be orange, but that will of course in every single cell only be the cause of one X chromosome, the other one becoming the bar body. Meanwhile, in the calico cat, they are heterozygotes for the black and orange. So some of their cells have the bar body is the black cell, and some of the cells, the bar body is the orange cell. And it appears that there are these patterns sort of appear where there are sort of random clumpings of cells that are all have the orange bar body and or all have the black bar body, and you get these interesting patterns that shows up on their fur, and that's essentially a statistical distribution of those cells for that cat. All right, we will talk about non-disjunction errors in the next video, but hopefully this was amusing for you, and I will follow this up with a couple of links and maybe some activities to do as well.